You may recognize our next speaker by name, not for the latest vaccine, but, in our community of course, for the compost method, Johnson Sue. Dr. David Johnson is a scientist and soil regenerator who's collected foundational data for us to understand the role of fungal to bacterial biomass as it supports plant growth. As a plant scientist myself, I was shocked to see that nitrogen, potassium, phosphorus did not best explain the amount of growth in the plants, but instead the community composition of the organisms is what created a beautiful correlation with plant growth. I was stunned. It was amazing. And, of course, as you will hear Dr. Johnson describe, his work has been pivotal to support the soil as a potential carbon sink to draw down the excess CO2 and sink it into those organic compounds in our soils. And with that, I will turn it over to Dr. Johnson.
This presentation is about regenerating the diversity of life in soils, hope for farming, ranching, environment, and climate. The influence of the microbes on this planet has been and is immense. We are the benefactors of everything that they have innovated, all the adaptations they have developed for surviving on this planet. Water came to this planet about 4.2 billion years ago. Bacteria showed up shortly after in the fossil record. One of the most novel adaptations on the planet, photosynthesis and oxygenic photosynthesis, developed about 3.8 billion years ago. This enabled the capture of energy from the sun, the ability to store it in a biochemical structure for use whenever needed. This adaptation completely changed the path for this planet. Virus precursors showed up 3.4 billion years ago. Nitrogen fixation about 3.2 billion years ago, and about 2.4 billion years ago, oxygenic photosynthesis developed. Another great adaptation that promoted the buildup of oxygen in the atmosphere. This oxygenation event played havoc on the dominant anaerobic microbes that led to the appearance of aerobic organisms, fungi being the first. As the oxygen in the atmosphere started to build up, an ozone layer developed, offering more protection for organisms on this planet. Mitochondria evolved about 1.4 billion years ago, and with this, the efficiency of energy use by microorganisms increased 18 times. The first single-celled animals appeared 750 million years ago, and land plants followed about 700 million years ago. Glomeromycota, the fungi that associate with plant root, helping the plant to access minerals in exchange for nutrients from photosynthesis, appeared about 500 million years ago. We just got here, but we are fortunate in that we get to enjoy all the adaptations that have come before us. This chart illustrates the influence that living organisms have had on this planet. The black line is atmospheric CO2 concentration. As you can see, it's been up as high as 7,000 parts per million over the last 600 million years. The blue line is the Earth's temperature. The red line, oxygen in the atmosphere. And these little clouds at the bottom, they're extinction events that have occurred during this period. We are here right now, about 14 degrees centigrade, 400 parts per million atmospheric CO2, and about 20% oxygen content. Each novel adaptation of living organisms over the last 600 million years has had an influence on this planet and its atmosphere. About 550 million years ago in the Cambrian era, there was an explosion of life in the ocean, with large population increases in the organisms that had calcium carbonate shell structures. Their activity, soaking up carbonates from seawater to build their shells, started to reduce atmospheric CO2 concentrations. Through the Ordovician, Silurian, and Devonian periods, Glomeraceae, mycorrhizal fungi, developed along with vascular plants, lignin, and coral reefs, all leading to another drop in atmospheric CO2, except for 2 extinction events that led to a small rise. The next novel adaptation was 8-meter-tall fungi in the Carboniferous period. Each step along this timeline, the processes for capturing, storing, and utilizing the energy from the sun get a little more efficient, and this period was no exception, as the majority of the coal we mine for producing steam or electricity came from this period. One of the reasons that plant carbon built up into coal seams was because there was no lignin-degrading microbes on the planet to degrade plant biomass. Lignin-degrading microbes came onto the scene about 275 million years ago, as well as gymnosperms. Part of the surge of CO2 as a result of lignin-degrading microbes and another extinction event. Conifers, evergreens, and angiosperms developed in the Triassic, Jurassic, and Cretaceous period, further decreasing atmospheric CO2. These are periods that the Earth's oil and gas reservoirs developed, so significant quantities of atmospheric CO2 were converted to living biomass and eventually oil and gas. The final drop in atmospheric CO2 concentrations began approximately 53 million years ago, with the global cooling period, land mass positioning changes, and when mammals, ungulates, and grasslands came onto the scene. This system placed a significant amount of atmospheric CO2 in the soil as 6-foot deep soil carbon profiles were developed due to the synergy between grazers and grasslands. Seeing that microbes dominated almost 80% of the time that this planet has been developing, we need to understand that microbes are the backbone of every organism and ecosystem on this planet. No organism can survive without its resident microbiome. Viruses are also an important component as they modulate the function and evolution of all living things. Approximately 800 million viruses fall on every square meter of the Earth every day, and 1/3 of the bacteria in the ocean are killed or attacked by viruses, adding to the flow of carbon and energy through living systems. You, as a human, have about 10 trillion human cells and about 90 trillion microbial cells. You also have over 1 quadrillion resident viruses. You have about 20,000 human genes. There are about 2 to 20 million microbial genes in your system, so you are a very small percent human and mostly microbial in origin. What they do for us is digest our food. They do cell renewal. They comprise what we thought used to be 60% of our immune system, but now it is more like 80%. They produce nutrients and synthesize vitamins we cannot get in our diet. They control our appetites and cravings. They detoxify carcinogens, prevent allergies, skin diseases, and asthma, turn on and off genes that regulate brain development, anxiety, depression, autism, arthritis, and our emotional behavior. So what happens when these microbial communities are disrupted through either administration of antibiotics or diet-related issues? We see autoimmune diseases. About every disease that we see in our society, from diabetes to inflammatory bowel disease, Alzheimer's disease, dementia, cardiovascular disease, all seem to be either directly or indirectly related to autoimmune systems, imbalances, and dysbiosis of our gut microbiota. Let's look at what happens in a diet-induced extinction of microbiota. This image illustrates what happens when you remove fiber from the diet of mice. As you can see, generation after generation after generation, the diversity in the stomach microbiome keeps decreasing. When you reintroduce fiber back into that diet, diversity does not come back. Generation after generation, you actually see a reduction in gut microbiota diversity. If we came back in with a fecal matter transplant from a healthy donor, you can see that gut microbiota diversity is restored immediately, and it remains generation after generation with the reintroduction of fiber into the diet. In our society since 1950, we have seen significant increases in multiple sclerosis, type 1 diabetes, asthma, and Crohn's disease. Crohn's disease is particularly painful for a lot of people. They can suffer for decades. They have surgical resections of their intestines, more antibiotic regimes to try to cure the disease. Many can die from it, and they're finding out if you can get a fecal matter transplant from a healthy donor, that in some cases Crohn's disease can be cured within 24 hours. Since the microbiome transfers work in restoring the human microbiome, can this work in agriculture? Because we know that we've destroyed the soil microbial structure with all the plowing, chemicals, and the fertilizer that we add. First, we have to look at how nature has set this up. There's a ladder of succession on plant systems that goes on between bare soil parent material up through your old growth forest. As you move up this ladder, you change in productivity from 90 grams of dry biomass per square meter up to 2,200 grams of dry biomass. What accompanies that increase in production is a change from a bacterial dominant system to a fungal dominant system, as well as going from a low carbon content to a high carbon content in the soils. Right now, we are here in our agri-ecosystems. Bacterial dominant, low carbon, and low production. Weeds grow very well. Weeds have a purpose on this planet. They are the organisms that begin to transition poor, broken soils up this ladder of succession and are good indicators of soil health. We need to be here in this range, about 1-to-1 on your fungal to bacterial ratio. That's where successional grasses, row crops, and vegetables do well. We also need to understand that agriculture is controlled disturbance. And in order to stay on that rung of the ladder, there's a certain amount of disturbance that has to occur. Nature developed a unique system about 53 million years ago. That's when ungulates and grazers evolved along with grasslands. They both appeared in the fossil record along the same period of time. Nature's system on the Great Plains involves bison. They were kept in tight herds by predators. They had to move around regularly so they could only graze about 40% of the forage that was there. The other 60% was trampled into the ground. They would deposit their dung, their urine, and their saliva. The dung would be rolled up in small balls like dung beetles and placed underground. Basically, it would be composted in place. Perfect temperature, moisture content, soil microbiota, and worm communities, allowing a maturation process. What was going on as the bison moved was a microbiological inoculation event that swept across the prairies. So, how can we mimic this system on agricultural land? Not everybody wants or can have bison or cattle on their property. My wife, Hui Chen, and I came up with a no-turn composting process that mimics nature. The process results in a fungal-dominant, biologically diverse compost. The bioreactor mimics what happens to dung and organic matter in the soils of prairies. Approximately 70% moisture content, no turning, doesn't freeze, and it's always aerobic. The bioreactor is up on a pallet, and we have 6 air columns up through the middle. The pipes are pulled out after 24 hours as the pile completely stabilizes itself with newly developed fungal hyphae and actinomycetes. Worms are added after the thermophilic phase when the temperature goes back down below 80 degrees Fahrenheit, and it takes about 1 year to reach maturity. During the composting process, it goes from approximately 160 degrees Fahrenheit down to 60 degrees. You can see the change in the population dynamics of the microbial community. It starts out very strong with bacteria from the phyla Firmicutes, and as they decrease in the compost, they practically go away. Others start small, like Bacteroidetes, and then come back strong in the finished compost. This slide shows a comparison of 2 composts made a decade apart, with completely different starting materials but using the same composting process. They are very similar in structure. The upper one is composed of shredded dairy manure and yard trash, and the one on the bottom was made from shredded alfalfa hay. Looking at the breakdown of the bacterial species as the compost matures, this is what the bacterial community looks like at 4 weeks. The diversity of the top 80%, there's about 23 species. This is what it looks like at 22 weeks. You have about 57 species in the top 80%. If you allow it to mature a year, there are over 99 species. So you see we have a 4 times increase in bacterial diversity as you allow this compost to mature. We started out with greenhouse trials, comparing our compost, the 2 here on the left, to 8 other composts available in our area. We observed the amount of plant growth, the blue bars on the graph, that the compost promoted. As you can see, our compost provided twice the growth of any other compost. Nitrogen, phosphorus, potassium are the key components that are supposed to indicate the potential for plant growth. It wasn't so in this test. Our compost was very low in nitrogen, phosphorus, and potassium compared to the other compost, yet we had over double the productivity. Well, we thought, okay, it's organic matter. Now, organic matter is important, but it did not correlate well to plant growth. We had measured the fungal to bacterial ratio along with the system physical chemistry, and we noticed the fungal to bacterial ratio was a very strong predictor of plant growth. This promoted a follow-up greenhouse trial with green chili to explore how fungal to bacterial ratio affected plant partitioning of carbon and nitrogen. We looked at what percent of photosynthate was partitioned into the root carbon, the brown piece of the pie, the shoot carbon, the green piece, and the fruit carbon. We also looked at new soil carbon, over and above what soil carbon we started with, the carbon that was respired, and as well the flow of carbon into nitrogen fixation. As we go from the bacterial dominant low carbon system here on the upper left to a fungal dominant high carbon system over here on the bottom right, we see a change in the percent of carbon flowing into the plant. In a bacterial dominant low carbon system, only 3% of the energy the plant captured through photosynthesis actually went into the plant. The rest of it went into increasing soil carbon, soil respiration, or through nitrogen fixation. As we move up the ladder to the higher fungal bacterial ratio, the amount of plant photosynthate increases from 17%, 21%, 30%, 37%, up to 56% of the energy captured in photosynthesis going towards the plant. We are here now in our agricultural systems, about 11% efficient. This may indicate that we should be able to increase our productivity on these lands about 5 times to reach the 56%. Another thing we observed in this greenhouse trial was the relative soil carbon respiration rate, or a measure of system carbon use efficiency. Notice in the bacterial dominant systems, low carbon, up to 55% of the available carbon in that system was respired. When you move towards fungal dominant systems, higher carbon mass as well, only 11% of the available soil carbon was respired, complementing the higher biomass production observed in fungal dominant systems was a significant increase in carbon use efficiency. After our greenhouse experiments, small plot trials were initiated. Inoculation rates for these trials started at 400 pounds per acre. So you can see on this white square, it's just a dusting of compost. So the compost was used as a microbiological inoculant, not a nutrient resource. This trial explored the potential of plant growth on desert soils, one that has no legacy issues from previous agricultural practices. Cover crops were grown on this soil, harvested and hauled off, and another crop was planted. This was to explore the capacity of an inoculated soil microbial community's potential to promote plant growth. On the left, you can see the beam, or the biologically enhanced agricultural management treatment, produced almost twice the biomass as the control. If you look in the background, you see how good the soil was. This is desert, so the soils are very poor. This is the second winter cover crop using the same seed mix where over 1,600 grams of dry biomass per square meter were grown. This is the third year's winter cover crop, 2,190 grams of dry biomass production and over 588 pounds of nitrogen fixed per acre, all with only a one-time application of compost, growing covers one after another and hauling off biomass production. Winter cover crop productivity increased from 492 grams to 2,244 grams in this 3.5 year experiment, demonstrating a 4.6 times increase in the productivity, very similar to what we observed in the greenhouse experiments. This winter cover crop is planted in October or November. It idles through the winter, about 2 inches high, and then wakes up mid-February, and it grows to the end of April. In 10 weeks, the plant growth equals the productivity of the most productive ecosystem on the planet when it has the right soil biology. The summer and winter cover crop together produced about 3,186 grams. Remember, the most productive ecosystem on the planet is 2,200 grams, so half again more than the old-growth forest. We also noticed that this system produced or fixed 777 pounds of nitrogen per acre per year. This is what the soil profile looked like when the experiment was started. All sand, no A horizon. This is our control. There's a 3-inch A horizon, and we saw a 7-inch A horizon in the compost treated. This chart represents findings from a metagenomic and metatranscriptomic analysis of the soils in the previous experiment. You can see the results in these 3 columns, where our control, our desert, and our compost treated. There are about 5 million different OTUs, different operational taxonomic units, representing about 2,750 species. 193 of these species came to the top and correlated well with plant growth. Grouping these species together and researching their function in the soil revealed approximately 21 of the species were either free-living or symbiotic nitrogen-fixing microbes. Another 13 were involved in nitrogen cycling or the ability to degrade nitrous oxide. Approximately 25 carbon cycling microbes. Carbon cycling is critical in these systems. It's like money in an economic system. If you don't have a certain velocity of money or carbon flow through the system, it's unhealthy. This carbon flow is synonymous to energy flow as well, so it's good to have these microbes in there to keep that energy flowing. 14 microbes were involved in metal oxidation, freeing up the elemental nutrients from the soil parent material, making them available to the plants as nutrients in exchange for plant photosynthase. 10 microbial species were involved in phosphorus solubilization. We have probably 40 years of phosphorus in agricultural soils from our year after year application, where most of the applied phosphorus is not available because of the physical chemistry in the soil. These organisms free that phosphorus up. There are multiple microbes that degrade plastics, pesticides, xenobiotics. These organisms can tackle just about any pesticide or herbicide that we produce. Phytohormone producing organisms as well. These organisms produce plant growth hormones that increase plant growth. Also found were microbes involved in quorum sensing. Quorum sensing is when microbes work together, expressing genes and metabolic functions as a community that they can't accomplish as an individual organism. Quorum sensing gene expression was in the top 21 genes expressed and demonstrated 6 to 10 times the expression level of housekeeping genes. Field trials were started in 2010. On the left, you're looking at our control. On the right is a biologically enhanced agricultural management treatment. There's a 4.5-year project looking at how fast we can increase soil carbon. Compost was applied only once at the beginning at a 400 pound per acre rate. From there, covers were grown year after year, mowed down, lightly disked in, and then the system was replanted so all biomass grown was left on the plot. After a year of being, the winter cover crop produced 250 grams of dry biomass compared to 50 grams of dry biomass in the control, a 5 times increase. 7 years later, on that same trial plot, you see 1,141 grams of dry biomass per square meter, which is almost 5 times the amount observed after the first year. This chart shows how the soil carbon increased over the 4.5 year period of this project. Soil organic carbon increased from 0.4% up to 1.5%, an annual increase of 0.24% in the top 30 centimeters of the soil. This rate is equal to 10.7 metric tons of carbon per hectare per year, or 37 metric tons of CO2 per hectare per year. This is a comparison of BEAM with other long-term carbon research projects. Most other researchers see only a 0.2 to 0.7 tons carbon per hectare per year potential to place carbon into the soils of agri-ecosystems. Promoting functional soil microbiomes promotes over 10 times that rate. We're looking at 10.7 tons. This is that 0.25% per year increase. We see the potential to do 19.2 tons, or 0.5% soil carbon increase per year. We included this soil in an experiment to explore the relative rate of respiration in 4 different soils: a desert soil, a conventional soil, that transitional bean soil, and an advanced bean. In a 1-year, 32 sample respiration assessment revealed a graph similar to what was observed in our greenhouse study. Comparing the production of conventional agriculture with bean systems, over twice the growth was observed in both cotton and chili. When you look at organic systems, they take approximately 30% hit on productivity because organic deploys conventional farming practices that use natural amendments in their approach. It's the microbiology that needs to be promoted in these organic systems to bring them up to what we're seeing in biologically enhanced agricultural management approach. Organics have potential to do much better. This is our cotton after 1.5 years adoption of bean, comparing a conventional approach here on the left at 180 pounds of nitrogen per acre to bean on the right with no fertilizers. There was almost triple the growth with the bean system. This is our cotton on July 17th on our bean plot. August 2nd, August 29th. 6-foot cotton, but that doesn't mean you have cotton unless you do. Our cotton production was approximately 5 bales per acre and over 3,000 pounds of seed per acre, an approximate $2,000 acre gross crop with no synthetic fertilizer usage. This is our chili crop, again bearing approximately twice the average chili yield in the Mesilla Valley. Farm-scale corn production field trials were next. 3 separate treatments were implemented on a half circle in Wilcox, Arizona. A conventional approach with 256 pounds of nitrogen per acre, a beam approach plus 15% of that nitrogen, or 38 pounds of nitrogen per acre, plus 2 pounds of the Johnson Sioux compost applied as an extract injected into the furrow at planting. And a third treatment, beam only, using 2 pounds per acre of the compost, again injected into the furrow at planting. This is what the results look like the first year. The beam plus 15% nitrogen matched the productivity of the conventional nitrogen approach and was $121 an acre more profitable. The beam only suffered a 2% yield loss, but was still $86 more profitable than the conventional approach. Looking at the nitrogen flow in these treatments, beam-only systems fixed 116 pounds of nitrogen per acre. The beam plus 15% nitrogen fixed 84 pounds of nitrogen per acre. And the conventional system leaked 122 pounds of nitrogen into the environment, the air, the soil, and our water resources. The following year, we implemented the same treatments growing pinto beans. In these trials, the beam plus 15% outperformed the conventional nitrogen approach with 11.7% increase in productivity, or approximately extra 5 bushels per acre, or $85 an acre more profit. The beam only outproduced the conventional approach also by almost 3 bushels an acre, and it yielded $47 more an acre in profit. It appears once you adopt beam and start cutting back on fertilizers, the soil seems to become healthier and more productive each year. Increasing soil carbon through implementation of beam practices helps rebuild soil water holding capacity as well. The first 1% increase in soil carbon increases soil water holding capacity 5 times. Implementing beam increases productivity and utilizes water more efficiently, so you get more crop per drop. Looking at the change in nutrient density of our food reveals lower and lower nutrient availability from 1940 to 2002. What can the adoption of beam in agricultural systems do to address this? In the first 20 months application of beam in our study plot, looking at 5 sampling periods, we observed a 1100% increase in the availability of water-soluble manganese and iron. Magnesium, calcium, zinc, and copper all increased from 83% to 40%, as well as nitrogen, phosphorus, and potassium increased 106% to 37%. Understand the pH in our soils is about 8.2. So iron and manganese availability is decreasing or declining due to the physical chemistry, but a functional soil microbiome helps alleviate these micronutrient deficiencies. The Johnson 2 bioreactor is a simple design. It can be made anywhere by anyone with little effort, and it takes about 2,000 pounds of wet material to fill it and yields about 700 pounds of finished material. The link for building, filling, caring for this bioreactor, and making the extract are the link below. Here are the results looking at 4 different application rates of the extract. The extract treatment ranged from 2 pounds per acre to 20 pounds per acre. As you can see the bar graph, there is no significant difference in productivity. What was noticed in a parallel experiment was the difference between injecting the extract into the soil, here on the right, or spraying the extract on top of the soil. The differences were dramatic. There was over double the plant growth when the extract was injected in the furrow as opposed to a topical treatment. A follow-up experiment was conducted on a summer cover crop harvested in the fall. The sorghum plant on the left had the extract injected into the furrow, and the one on the right had the topical extract treatment. The in-furrow treatment reached reproductive maturity, where the topical treated failed. The difference between these treatments was a successful crop and a crop failure. These are results from a farmer in Belgium using a seed treatment of the Johnson 2 compost on a multi-species cover. He saw exactly the same results that we saw, a doubling in productivity on his crop. Again, in our use of compost as a microbial inoculant in cropping systems, we are trying to mimic what's happening in the natural grazing systems. Let's briefly look at how this is implemented in adaptive multi-climatic grazing management systems. These are results from Gabe Brown's ranch. He has increased his soil organic carbon from 1% to 6.5% in 20 years. For the first 13 years, from 1993 to 2005, no-till, crop diversity, and cover crops enabled Gabe to achieve an approximate 1/10 of 1% increase in soil carbon per year. When he stopped using fertilizers and he reached 3% soil organic matter, he started using multi-species covers and eventually livestock integration. This rate of soil organic carbon began to increase to approximately 0.5% soil carbon increase per year. This is what his pastures looked like when we visited him. For the previous 3 years, he had 5 inches of rain per year. It's not a lot of rain, but his pastures had forage that grew over the hood of his 4x4 truck. Gabe has relayed that he's seen up to a 30 times increase in the productivity of his pastures. This is Will Harris's operation in Georgia. Will has increased his carbon from 0.75% to 8% in the last 20 years. This is a soil organic carbon increase of 0.36% per year. We were able to get a hold of some respiration data off of 5 of Will's pastures, looking at different plots he's transitioning from 1 years to 20 years. The respiration trend line mimics the greenhouse experiment and field experiment conducted on the beam transition trials. In a project with Peter Bick at Arizona State University, we are comparing adaptive multi-paddock grazing managed ranches to continuously grazed ranches in the southeastern U.S. Our portion of this research was to compare the influence of these two approaches on soil biology. The AMP systems demonstrated an increased photosynthetic capacity of 93.1 grams of dry biomass per square meter. The AMP systems almost had 0.5% higher soil organic carbon percentages. And as well, AMP systems demonstrated a 35% decrease in the relative rate of respiration, or increased soil carbon use efficiency when compared to continuously grazed systems. Practicing the grazing management that mimics the bison on the Great Plains helps to regenerate the microbial community population, structure, diversity, and biological functionality of these grazing systems, increasing photosynthetic efficiency on the front end, reducing the carbon flow out of the system on the back end, and these two attributes together help increase soil carbon. There is now a push to implement carbon capture in the soils of agroecosystems. The carbon we put back into the soil needs to be a biologically functional carbon, carbon in the form of microbes that fix nitrogen from the atmosphere and furnish it to plants, and also microbes that break down the soil parent material to gather essential elemental nutrients and shuttle them to the plants in a form of biological barter in exchange for photosynthetes that contain sugars and proteins. Our current greenhouse gas emissions from burning fossil fuels are approximately 3% of the total amount of CO2 flowing into the atmosphere. The other 95% of CO2 that flows into the atmosphere results from respiration from microbes in the soil, our ocean, and respiratory processes from plant metabolism. We have a choice now. Do we try to make large changes in a small component of the system, for example, drastically cutting our fossil fuel consumption, or do we make small changes in a large component of the system to improve the carbon capture potential of our soils? Globally, there are approximately 1.25 billion hectares of temperate grasslands, similar to the AMP ranches we studied in the southeast, and another 1.36 billion hectares of arable farmland, as I described in the first half of this presentation. Our current soil systems are broken. We will not be able to accomplish carbon offsets of any magnitude in the soils of our agroecosystems with a business-as-usual approach. What is necessary is to adopt practices that offer improvements in efficiency for increasing photosynthetic capacity, to increase the amount of carbon flowing into the system, and those that improve carbon use efficiency on the back end to slow the flow of carbon out of the system. What amount of carbon offsets could our previous observations offer if adopted on these lands? We could achieve an extra 18.2 billion tons of CO2 pulled from the atmosphere through increases in photosynthetic capacity and improved carbon use efficiency using the data from our observations on the 1.25 billion hectares of rangeland on this planet. Another 27.5 billion tons of CO2 could be pulled from the atmosphere through increases in photosynthetic capacity and improved carbon use efficiency on the 1.36 billion hectares of arable land on this planet. Besides for providing a viable path forward for reducing atmospheric CO2 concentrations, the adoption of regenerative practices will also provide the potential to restore degraded lands and countries across the globe, restore soil water holding capacity, helping to reduce downstream flooding while promoting improved plant water use efficiency. help remove carbon dioxide and nitrous oxide from the atmosphere, and use them as nutrients, not pollutants, reduce methane emissions, and also promote growth of microbes that remove and metabolize methane from out of the atmosphere. For those of you who are saying we can't do this here, this is Alejandro Carrillo's ranch in the Chihuahuan Desert in northern Mexico. When he took over management of this ranch about a decade ago, it took him 250 acres to support one cow. He normally gets 6 to 7 inches of rain on a good year. He started practicing adaptive multi-paddock grazing on his rangeland. And now he's changed that environment completely, from a desert back to a grassland. Now it only takes about 20 acres to support one cow. So with proper grazing, he's increased in photosynthetic capacity 10 times. This used to be a grassland hundreds of years ago. You can see a 3-foot deep soil carbon profile. But overgrazing destroyed these systems and desertification took over. Another example, Di and Ian Haggerty are farmers in Western Australia. They farm about 20,000 hectares of dryland barley, and they have stopped using fertilizer and are applying the microbiology, using compost as an extract injected into the forward planting. It's at the same rate of application that we do, about 2 pounds per acre rate. On their barley crop, they do about 3 tons of barley per hectare in a dryland farming area that conventionally gets about 1.5 to 1.6 tons per hectare. You can see the rhizosphere develop on the roots of the barley seedlings. What is most interesting to me is the picture on the left. They have already harvested the barley in this field, but with a late rain after harvest, you see a C4 grass sprouting. This grass had not been seen in over 4 decades, sprouting up in nice, neat rows, exactly where they had injected the compost extract when they planted the barley. Application of these microbes seems to reawaken the soil seed bank. This is another interesting thing we came across on Facebook, and this is also from Australia. A rancher sprayed some of the Johnson Sioux compost extract on his rangeland. He could only spray on one side of this tree as the branches were too low on the other. It's quite astounding to see that one side of the tree leafed out like that, and it leafed out almost a month before the other side of the tree did. And this is the difference in the chestnuts between treated and untreated on the right. So we can meet the challenges we face. We can rebuild degraded soils and increase soil fertility. We can produce more nutrient-dense food. We can use less water to grow crops by increasing both water storage capacity in our soils and plant water use efficiency. We can reduce our energy use by allowing nature to extract or fix essential nutrients. We can reduce agrochemical pollution and atmospheric CO2 at the same time, but most importantly, we can improve farmer and rancher productivity and profitability. Why should we pursue this? There is no Planet B. I would like to thank the Howard G. Buffett Foundation, the Thornburg Foundation, the Globetrotters Foundation, and the No Regrets Initiative, the Mighty Arrow Family Foundation, and the Laird Norton Family Foundation for making this research possible. Here are some links you can get for more information at the Center for Regenerative Ag and Resilient Systems at California State University at Chico.
Spring is in the air, and there's never been a better time to launch your career in soil regeneration than right now. The Soil Food Web courses are being offered at the lowest ever price of just $2,900. That's an incredible 42% off the regular price of $5,000. Whether you're a farmer, rancher, market gardener, or just someone who's passionate about the planet looking for a way to make a big impact, this could be for you. Here's what some of our students are saying about the Soil Food Web training program.
I find that this information hasn't been taught to me, and I had to get off my high horse. And even though I have a PhD, I feel like I'm totally undertrained.
I feel like I'm learning more with this program than I have with in-person classes in the past.
I've taken classes on microbiology before, but this course really makes a difference in the way that a story is put together that unveils the relationships between plants and all those beneficial organisms that we just cannot see without a microscope.
If you're looking for something that does a deep dive into soil biology, this is it. It is just an incredible knowledge base and is really relevant to what's going on right now in the world.
Without it, the only way I could have gained this knowledge would have been by dropping my life and going to graduate school. And that just wasn't realistic for me. But Soil Food Web has made it possible for me to build a meaningful career in land restoration.
There was a payment plan, which meant that I didn't have to come up with huge amounts of cash up front.
I was really nervous. I was going to put quite a bit of money down and not get that bang for my buck. But once I actually got into the FC courses, I was incredibly impressed with how professional they are and actually the level of education you receive. This is the career path I've been looking for in the biological community and I was having trouble finding.